Hello, my name is Giselle Haddad, and I am going to be presenting my research on the role of TREM2 and CD33 in Alzheimer's disease. A little bit about me. I am a senior at Bloomfield Hills High School in Bloomfield Hills, Michigan, which is where I am currently presenting from. And my mentor for the Polygence research project was Tatiana. I had an overall great experience with my mentor, and I was very well supported throughout the entire process. My interest in Alzheimer's disease stems from previous computational research that I've conducted on both Alzheimer's and Parkinson's disease. I identified potential biomarkers that can be used as treatment targets for both disorders, and I was motivated to continue research on Alzheimer's after attending a virtual session given by a representative of the Alzheimer's Association, where she emphasized the need for further research on Alzheimer's disease and the importance of helping patients who are experiencing this disorder. So I'll start by providing some basic background on what Alzheimer's is and the symptoms, risk factors, and treatment options that are associated with it. So Alzheimer's disease is a multifactorial neurodegenerative disorder characterized by the accumulation of amyloid plaques and neurofibrillary tangles. I will go more into detail with those concepts on the next slide. It is the leading cause of dementia, impacting more than 47 million individuals worldwide. Symptoms associated with Alzheimer's include difficulty with memory, cognitive understanding, behavior, and daily functions, and some risk factors are depression, smoking, alcohol use, and various environmental factors. Currently, inhibitors, memantine, which slows neurotoxicity, and antibodies are used to treat Alzheimer's disease. No direct cause has been identified, so personalized treatment is currently unavailable. This image provides a visual depiction of what I was describing in the previous slide, and I've also provided a definition of both amyloid plaques and neurofibrillary tangles here. I would like to mention that the amyloid plaques form between brain cells, which is what prevents communication and causes inflammation and cell damage. And that ultimately leads to the neurodegeneration and Alzheimer's brain that we see in this image. My thesis for both my paper and this presentation is that the upregulation of TREM2 and CD33 is associated with the development of Alzheimer's disease. I started my research process by accessing the Gene Expression Omnibus, which is a database for gene expression profiling that provides datasets studying Alzheimer's disease. An extension of this database is GO2R, which is used to examine the differences in expression among genes. I then accessed the KEGG database, which is composed of biological pathway maps, and I used the string database, which contains maps of known and predicted protein-protein interactions. So I selected two datasets from the Gene Expression Omnibus, and they both studied Alzheimer's disease. One of the datasets studied the disease in humans, and the other studied it in mice. The samples from each dataset were organized into two groups, Alzheimer's disease and healthy control. After organizing my data, I input it into GO2R, and various diagrams were produced, one of which is the volcano plot, which displays statistical significance or p-value versus magnitude of change or log-fold change. P-value is the probability that results from data are due to chance. We want a lower p-value to have statistically significant data, and the cutoff for that is a p-value less than 0.05, which is represented by all of the colored dots, both red and blue, in both of these volcano plots. Log-fold change is a statistical value that is used to determine whether a gene is upregulated, meaning overexpressed, or downregulated, meaning underexpressed. Upregulation is represented by positive log-fold change, and downregulation is represented by negative log-fold change. The blue dots in these volcano plots represent genes that are downregulated in Alzheimer's disease, and the red dots represent genes that are upregulated in Alzheimer's disease. Another plot that was provided from the GO2R analysis is the UMAP, which shows similarity between samples. And in both of these plots, we see a general differential distribution of samples between Alzheimer's disease and control groups. Displayed here are the statistical values for TREM2 and CD33 from the datasets. Based on the information provided in the previous slides, the values here show that the genes are differentially expressed and upregulated in Alzheimer's disease patients. Before we proceed with the rest of the slideshow, I'd like to provide a brief description of both TREM2 and CD33, and I'll start with TREM2. Which is a triggering receptor expressed on myeloid II cells, which are a type of white blood cell that originate in bone marrow. And TREM2 is expressed throughout the central nervous system, primarily in white matter. CD33 is a protein coding gene that stimulates phosphatase and sialic acid binding activity. So I'm not going to go too far in depth with the KEGG pathway analysis for sake of time, but I will say that I identified that TREM2 is involved in osteoclast differentiation, which is responsible for bone resorption. And highlighted in purple on this diagram, you can see where TREM2 is expressed in the pathway. I also found that CD33 is involved in hematopoietic cell lineage, which produces blood cells from hematopoietic stem cells. And again, highlighted in purple in this diagram is where CD33 is expressed. So after looking at the KEGG pathway for both genes, I inserted each of them into StringDB, which produced these string maps. And I found that both genes are involved in regulating tumor necrosis factor production and the negative regulation of cytokine production. Tumor necrosis factor is a cytokine, which is a protein affecting the immune system, and the negative regulation of cytokines prevents their production and inhibits the regulation of the immune responses. So after conducting this research, I reviewed previous studies and found that TREM2 rises with increased levels of beta amyloid, which is a major component of amyloid plaques. And they also found that increased expression of CD33 results in decreased phagocytosis, which is a process where the cell engulfs particles and other substances. This ultimately limits the uptake of amyloid plaques and results in the development of Alzheimer's disease. In terms of the clinical impact, research suggests that increased and regular exercise will regulate TREM2 expression in the brain, and this was confirmed by a mouse model study. It was also stated that treatment targeting CD33 downregulated pro-inflammatory activation genes, ultimately reducing Alzheimer's disease pathology. So after conducting all of this research and review, I found that one of the major limitations is that gene therapy can have unknown side effects. It can cause errors in the genetic code of individuals, and it can result in unwanted immune responses. And in an effort to prevent these issues from occurring, it's essential that we continue conducting experiments to confirm the efficacy of gene therapy as a treatment approach. And it's necessary that we specify experimentation to focus on TREM2 and CD33. It's also essential that we participate in events that advocate for Alzheimer's disease awareness and fundraise for organizations like the Alzheimer's Association and the Bright Focus Foundation, which both aim to advocate for Alzheimer's awareness and raise money for research. In an effort to have my own personal impact in the world of neurology and to advocate for more awareness to these neurodegenerative disorders, I formed the Neurology Youth Advocacy Association, which is a nonprofit organization that aims to raise awareness on neurodegeneration and brain health. We will do this by instilling a devotion to scientific research, outreach, and service in younger generations. I have a link to the website and our Instagram to the right if you are interested. Thank you so much for listening to my presentation. If you have any questions, you can reach me via email. And these are my references. Thanks again.